Today, I'm going to give a review report from the TgX DDI transcriptomics biomarker working group. Mostly, I'm going to focus on the status of these biomarkers qualification process. And these are members of our eSTAR TgX DDI working groups. Carol Yak and I co-chair this working group, and we have partners showing at the right panel. And the TgX DDI biomarker was developed to address the issue of the high positive rate of the standard genotoxicity test assays. And we know those test methods are sensitive, but lack of specificity, which lead to sort of like issue of the misleading positive results. And also those methods that do not provide information on the mechanisms. So we need to have the sort of like a novel assay or method, which is compatible with the high throughput platform, and also provide mechanistic information in the human relevant cell culture models. Next slide. The TgX DDI is the biomarker that we developed to address this issue. We actually identified this biomarker first using the human cell line TK6 cells, and the purpose is to predict the DNA damage inducing required DDI chemicals. The learn set was from a transcriptomic profilings of 28 prototype DNA damage inducing and non-DNA damage inducing chemicals. And using the machine learning approach, a biomarker comprised of 64 genes was identified and are being used to predict of the DDI potentials. And here are some publications for the biomarker identification and development and validation. We publish in EMM and PNS, and also the use of this biomarker with metabolic activation system has been developed by Carol Yak's lab. Next slide, please. And this is basically, that was the first stage of this transcriptomic biomarker. The longer vision for this transcriptomic biomarker development started from identification of the biomarker and then do validations, and further on, the dose response model, even in vivo, either preclinical or in the patient, need to be done, and then eventually goes to the level of regulatory decision making. And please, next. As shown here, those are different stages. Like we are really at the first stage by now, but then following up, we can reach the threshold of toxicity, understanding on that, and transcriptomic point of departure. And then wrap it up, eventually, hopefully by going through tier 1, 2, 3, we will be able to go to the context-specific regulatory applications. Thank you, next slide. And this context of use was published in our PNAS paper. Is really just showing the application of this biomarker in in the left, panel A showed in the pharmaceutical drug development process that we proposed, and then panel B shows how it can be used in the industry or environmental, chemical risk assessment process. It's like in panel A for the drug development, we will come down through the standard genotoxicity assays, those 3. And then if there is, or potentially there's a chance to have the misleading positives in the DNA, chromosome damaging assays, then TgX DDI will be able to help de-risk this irrelevant or misleading positive. And similarly, this can be used for the in panel B, for the chemicals in the with the environmental issues. Next slide, please. And after the identification of this biomarker, we have done extensive validations. Up to date, we have tested about like 100 agents, and the figure showing in this slide was from one of the publications doing these validation studies, and it basically showing this as an example. The TgX DDI can sensitively detect the positive genotoxicity agents as shown in the class 1, which are known direct acting DDI agents, and it will call negative for the known non-DDI agent, which shown in class 4. And most importantly, it can be used to de-risk the misleading in vitro positives, which are class 5. Out of these 11, like misleading in vitro positive chemicals, only one came up as a DDI positive call by TgX DDI. The rest of them are all predicted as non-DDI, which is consistent with their in vivo genotoxicity results. Next slide, please. So basically, this showed other like case studies that we have done so far. They are all in publications now. The first one is de-risked the misleading positive caffeine as an example. And secondly, we identified genotoxic hazard in chemical screening and evaluate concordance with upper endpoints for those drugs. And third study is a de-risk agent with a questionable genotoxicity findings. And the data showed high concordance with the high throughput comet assay. And our assay was run on 2 platforms. Next slide, please. Here are some recent advances in TgX DDI studies. This recent published paper in LTEX showed the high throughput and highly automated assay developed using the nanostring PlexSat technology. And next one. And this another recently accepted paper in Frontier of Toxicology, reporting this case study integrated genotoxicity testing of 3 anti-infective drugs using the TgX DDI transcriptomic biomarker and high throughput comet chip assay in TK6 cells. Next, please. And our biomarker has been discussed and as a representative in vitro biomarkers in the recent AIDS International Workshop on the genotoxicity testing. And here are some consensus statements come from this workshop. And we believe that transcriptomic biomarkers for genotoxicity can be used in a weight of evidence evaluations to identify potential genotoxic mechanisms and hazards, to identify misleading positives with in vitro genotoxicity assay, and as part of integrated testing with standard battery of genotoxicity tests and or new approach methodologies. And several biomarkers have been developed and validated and have demonstrated performance in multiple laboratories. These biomarkers can be used following prescribed study designs and models designated through existing validation exercise in the weight of evidence evaluation. And this is a tool developed by the National Toxicology Program. It's an online interactive tool, so it can actually facilitate the use of this biomarker by by different users and clients. So hopefully it can widen the application of this biomarker. Please, next. So far, as I showed all those studies and our previous publications that's showing here, it has been a long way. And here when we have gone through the developed method, define context of use and validation and proof of concept, and followed by the case studies that's showing here. And in parallel of all this long journey of continuing studies and publications on TgX DDI, it's also a long process for the effort to pursue the regulatory acceptance. We actually studied over 10 years ago for the first meeting to talk about this biomarker qualification. And now, the up-to-date, I'm going to talk more in the next slides, but just to brief, we are now actually already in the FDA biomarker qualification plan, and then in the accepted study going on. And also, very importantly, this TgX DDI biomarker has become part of the Health Canada's Genotox 21 platform. And we are planning to propose this biomarker in the integrated approach to testing and assessment IATA in the OCED platform. Yeah, we can go to next slide. Thank you. I'm going to talk a little bit more on the FDA biomarker qualification process. And these are 5 steps that for a biomarker qualifications. And we so far, as you can see here, we have completed the first 3 steps, and we are now in the process of conducting of the final validation studies. Next, please. Yeah, and we are excited to let you know that HESI our working group, was awarded a $250K grant from the FDA in this May. And this award is to support the multi-site ring trial, which is requested by the FDA for the final validation study. And this funding will support materials and logistics, and then also the PI support at Georgetown and some of the management support at HESI. Okay, and this UO1 grant is led by Dr. Cyril Petit at HESI, and this study design has reflected years of conversations and input from FDA and biomarker program reviewers. If I have to say any lessons we learned from this long journey, well, it's not done yet, but the first thing I need to say is do not give up. It has been many, many turnarounds and improvements and communications going on. Please click the next, yeah. So the validation study design reviewed by FDA and agents' feedback has been incorporated into this plan. And then the objective is to assess the cross-laboratory reproducibility of TgX DDI classification course involving one technical platform, which we choose to use is a NanoStream platform. And it's going to be run at 4 sites with 14 chemicals plus controls. And the plans involved the independent laboratory site will receive the test compound, and then we will test the 3 concentrations with the vehicle controls. And then cells will stick with the TK6, samples will be in triplicate, and then all this experiment will be carried out, follow the SOP. And the analysis and classification will use a the standard, software tools, which I have shown you in the previous slide. Yes. This basically to meet the requirement of the FDA, BQP program, we need to have 4 additional partners, because they ask us to have at least 4 independent laboratories. So to do that, this showing our partners get involved in these final validation studies. In addition to our previous members, now we have 3 more companies are joining us, as shown in kind here, and their role for the multi-site are showing at the in the table at the right-hand side. Yes. Next, please. And our next step will be to complete the validation study, and then we will submit the final qualification plan to the FDA, and then we will organize the data and publish results in peer-reviewed manuscripts. Potential future work included the OECD IATA validation, and the workshop highlighting this qualification path. And we also would like to broaden the application of this biomarker beyond the weight of evidence approach. And thank you. That's the end of my presentation. And if I miss anything important, please, like Carol, Sadaf, and Cyril, just come in and and also any question from the audience.
Great, thank you, Henghong. Yeah, there was some support from Carol in the chat discussing the expert international gene tox community as evidence at the IWGT meeting that took place a few months ago. And she wrote that TgX DDI experiences were really informative in that group's consensus statement. And that was chaired by Roland and Chris Corden, and you were the rapporteur. So any questions or comments from the group for this work? Feel free to raise hand or put in the chat.
We have
maybe. Okay, yeah. I was gonna ask, is there? Oh, Cam has his hand up. I'll ask mine real quick. This has been a long process. If we were to start this today, is there a recommendation you would make?
Definitely. I think it's during those process we learn a lot. First of all, this is transcriptomic biomarker. It's a new concept in the field. So it's new for us. It's also new for the regulatory agent. So it's at the beginning, I don't think they were very clear on what they would like to see from us. So it's a lot of back and forth. But I think to now, up to date, I think it's like they have a clearer pipeline for that. That was helpful. So and then of course it still need a lot of communication to see exactly what they would like to hear, to receive from us. Another thing is always, I think it's always good practice to keep everything in sort of like standardized pipeline, even at the beginning of the identification of the biomarker, like all the algorithm and statistical methods, and try to use very standard method, and then keep every single step. Sometimes they want to see your footprint. So that's another thing. I guess it's what, if you are talking about the lessons we learned, yes, I think there are definitely more. I mean, like Carol and Cyril has been really involved a lot in dealing with the FDA and talking with them. And so I guess I answered some of your questions.
Yeah, no, that's really helpful. And you'll hear maybe later today in our carcinogenomics group, that's a similar topic that, as we're beginning to develop biomarkers. So I think any learnings from your all's work is really going to be useful. So Cam's up next. Cam, you should be able to unmute now.
Great, thanks. Yeah, really good work, great work. I mean, it's very persistent as well. So 10 years, a long time. I also want to congratulate you on your grant. It's really super.
Thank you.
A couple of questions. So are these molecules in this validation set for this new study, are they the same as the validation set that you used in your publications? No.
No, that's actually is one of the consensus we got in this the recent workshop. We suggest when you are developing the biomarker, like the learn set, the learning set, the compounds you use in the learning set should be different from the one you use for the validation set. And then of course you can have part of your learning set chemicals in your validation, just basically to show the assay, the reproducibility of the results. But then the really validate the power of prediction for the specificity, all this has to come from the totally different validation set. So the answer to your question is validation, compound library, or the set, they have, some of them do exist in the learning set, but majority is not.
Right, thanks. And then just one other question. So we've used this biomarker a bit in our lab, and we seem to have always had to have a very robust gene expression response in the transcriptome. So something like 1,000 differentially expressed genes before this biomarker becomes positive, for molecules that are known positive. Is that a sort of a general observation that you actually have to have a pretty robust biological response before you see this biomarker light up?
You mean robust means like how much it's the change of the gene, or
yeah,
how
just robust
wide?
from a number of differentially expressed genes. We had to have
The number
hundreds.
of,
Yeah,
so
in addition to
talking,
your biomarker.
yes, right. Well, it depends. For most of the chemical we are testing, the biomarker is only represent a small portion of the differential gene expression. Those are sort of like the differential genes in common for this group of the DDI agent, right? Because the reason is that they're, in addition to the DNA damaging induced response, there are actually other other mechanism of action also involved. They may not have any common between different drugs we are using in this first of reason. And secondly, even just for the DNA damaging inducing genes, this only the tip of the iceberg. The reason we pick them is because they popped up. They are popped up like off the ocean surface, but they are actually a big chunk of them, maybe with less less induction. I mean, I'm talking about log ratio or fold change, less reduction, or they can be not so uniformly induced or repressed in different compound in the DDI. Like some of them may have stronger induction, some of them may not. So those were not selected by the machine learning algorithm.
Thanks, Henghong. Thanks, Cam. Yeah, there's a comment from Mark in the chat. Mark was saying that it would be great to put together a list of do's and don'ts that could be shared with any group that would want to submit a biomarker. Henghong, I know you had suggested, it was in the slides, maybe some kind of workshop. I wonder if some kind of short, I don't know if there'd be a publication or what would be helpful.
We are actually working on the publication. We'll put this consensus together into, I think it's going to be a special edition from a journal. I'm not sure which one, but yeah, it definitely will be published. We're working on that.
Great. And then Carol also noted that reproducibility is often a challenge with omics work, as we use many pipelines. The IWGT proposed an omics reporting framework module for the OORF that Mark Viant presented briefly for the omics biomarkers. So that's something too we've again talked about for the carcinogenomics project, maybe adopting that template. And I know, Carol, you had discussed that there's maybe going to be one that's going to be specific for biomarkers. So I think like that reporting, as Henghong noted, is really important, so we can know what we did and if we have to go back and show any step. So I appreciate those comments. I don't know if you have anything to add to that, Henghong.
Yeah, I think all these are great add-ups to my answer.
Great.
Yeah.
Well, I don't see anything else. I really appreciate this presentation. I think there's going to be exciting work to share next year as things wrap up. So I really appreciate your presentation, and thanks to the whole team for this work. Thank you.
Thank you.
Oh, I'm sorry. If maybe before you go, there was one last one. If you're still there, hang on. Have you tested any high production volume chemicals or those on the TSCA work plan? So more industrial chemicals.
First of all, I'm not quite sure. I know exactly what the high production volume chemicals mean. You mean like the commonly used in the, you know, like in industry or something?
Yeah, it's a designation more under EPA, I believe. So it's, you know, not pharmaceuticals, not pesticides, but those used in the industrial context.
Well, I don't think we have focused on that. I mean, if like one or two happen to be in this category of chemicals, it's possible, but we didn't really like try to address this class of chemicals. I don't know, Carol, do you know any of the, you know, chemical we tested belong to the high production volume chemicals?
I don't think they're high production volume chemicals that we've tested, but we have tested chemicals on Canada's domestic substances list. And we have tested now 10 chemicals on the new substances assessment and control in commerce list. So these were all data poor chemicals that you know, needed some genotoxicity information. So those were some of the case studies that we did there, but not high production volume chemicals to my knowledge.
I do remember one of the chemical in our validation, you know, in that PNAS paper, it was something we use actually either in perfume or the lipsticks. I just happened to notice that, you know, it's a familiar name to me in the cosmetics field, but that's pretty much it.
Yeah, maybe Santinder, you could send that list, and that could be something of further interest to maybe expand the domain.
Sure, yeah.
Great. Well, thanks, Henghong. Thanks, Carol.